Good morning, we are from Group 2. We'll explain about new drug development. First, what is a drug? A drug can be defined as any substance that when taken into the body can induce a change in an organism's physiological states. These alterations can manifest in multi ways. A drug might subtly influence our mood or perceptions, or it could have more dramatic impact on our body functions. It is important to note that drugs can have different origins. Some drugs are maybe from natural bounty, such as aspirin, which can be extracted from willow bark, or lithium, a naturally occurring minerals. On the other hand, many drugs are synthetically produced in the laboratories. These synthetic drugs may mimic the structures and function of naturally occurring compounds or represent entirely novel creation. Another crucial distinction lies between drugs and food. While both influence our body, their primary purpose is different. Foods serve the cornerstone of our well-being, providing essential nutrients for growth, repair, and maintenance of body functions. Drugs, however, induce specific change beyond basic nourishment, and they target a specific physiological or physiologically process that produce a desired effect.
Excuse me, doctor. My name is Louise with student ID 3017, and I'd like to explain about the drugs classifications. So, drugs can be categorized based on various factors, including their legal status, how they are obtained, and also their intended use. Here's a breakdown of some common classifications of drugs. First is the prescription drugs. These require a doctor's prescription due to their potential for misuse, complex administration, or powerful effects. For example, antibiotics, painkillers, and also antidepressants. Next, over-the-counter drugs, or OTC. These are readily available for self-care of minor conditions without any prescriptions, such as pain relievers, allergy medications, and also antacids. Next is illicit drugs. These are illegal to possess or use without a medical license due to their high potential for addiction and also harmful effects. For example, heroin, cocaine, and also marijuana. And lastly, complementary and alternatives for medicines, or CAM. This category encompasses a wide range of products and practices used alongside conventional medicine, and sometimes with limited scientific evidence to support their efficacy. For example, herbal supplements, vitamins, and also acupunctures. That's all from me. Thank you.
Okay, next I will be explaining on how do drugs work. Drugs exert their effects through specific mechanisms. They don't simply change our state of being in an undefined way. Instead, they interact with specific molecules within our bodies, often proteins or receptors located on cell membranes. These interactions can have various consequences. Antagonists act like gatekeepers, occupying receptor sites and preventing natural molecules from binding and triggering their usual response. Agonists, on the other hand, mimic the effects of natural molecules by fitting into receptor sites and activating desired cellular processes. Additionally, some drugs function as enzymes, accelerating and or inhibiting specific chemical reactions within the body. That's all for me. Thank you.
Excuse me, Doc. My name is SAILIHIN with the student ID 3014, and now I'm going to be explaining about what is new drug development. So, new drug development can be defined as the comprehensive process of bringing a novel medication from its initial conception to widespread use by patients. This intricate endeavor involves a multitude of steps. The first crucial step involves identifying a specific molecule or pathway that plays a role in the development or progression of particular diseases. Once this target is identified, researchers embark on a quest to discover potential drug candidates that can interact with the target and produce a desired therapeutic effect. These promising candidates are then meticulously optimized to improve their potency and selectivity. The next phase involves rigorous testing. So the first is preclinical studies using animal models to assess safety and efficacy. and then progressing to meticulously designed clinical trials in humans. Following successful completion of clinical trials, the data is submitted to regulatory agencies for thorough review and approval before the drug can be manufactured and made available to patients.
Excuse me, Doc. My name is ZAKHRUFULLAYLI ALISYA FAZZA with the student ID 3016, and now I'll be explaining about the importance of new drug development. As we all know, new drug development is a complex and lengthy process, yet it is vital for several reasons. First of all, it equips us to combat the ever-evolving challenge of diseases. New pathogens can emerge, and existing ones can develop resistance to current medications. Therefore, continuous research and development are necessary to discover novel antibiotics, antivirals, and therapies to combat these evolving threats. Second, new drug development is imperative for addressing unmet medical needs. Millions of people worldwide suffer from chronic illnesses, but current treatment options might cause undesirable side effects. New drug development strive to create more effective medications with improved tolerability. And lastly, new drug development holds immense promise for unlocking the potential for groundbreaking treatments. The concept of personalized medicine offers the possibility of tailoring medication to an individual's unique genetic makeup, potentially leading to improved efficacy and reduced side effects. Gene editing therapies delve even deeper, with the potential to correct faulty genes at the source of disease, paving the way for permanent cures for genetic disorders.
Stage 1. Drug discovery. The discovery phase is the foundation for new drugs development. Here, scientists embark on a trialing quest to identify potential drug candidates. This phase involves a multifaceted approach, including pinpointing a specific target molecule or pathway that plays a role in a particular disease. Once a target is identified, research leverages high-throughput screening, a powerful technique that allows for rapid testing of vast libraries of compounds to find ones that interact with the target. Finally, the most promising compounds are meticulously optimized to enhance their potency or selectivity, ensuring they specifically target the disease process while minimizing side effects.
Next is stage 2, preclinical development. After potential drug candidates are identified in the discovery phase, they progress to preclinical research, a critical stage that bridges the gap between the laboratory and human testing. In vitro testing, meaning in class, involves evaluating the drugs in cell culture to assess their activity and potential for adverse effect. If the results are encouraging, the drugs advance to in vivo testing, meaning in the living organism, where they are administered to animals to determine their efficacy, safety, and pharmacokinetics. It explains how the body absorbs, distributes, metabolizes, and excretes the drug. This data is then meticulously compiled and submitted to regulatory agencies such as the Food and Drug Administration, or FDA in the United States, for approval to proceed to the next crucial step, which is clinical trials in humans.
Excuse me, Doc. My name is KAYSHA, student number 2012. Now I'll be explaining the third stage of drug development, which is the clinical development. So clinical trials represent the real test for potential new drugs. Here the drugs are meticulously evaluated in humans to assess their safety and also the efficacy. Clinical trials are meticulously designed and phased. Clinical development is divided into 3 phase trials. The first one, which is the phase 1 trials, they involve a small group of healthy volunteers to assess the drug's safety, appropriate dosage, and potential side effects. And if the drug demonstrates an acceptable safety profile, it advances to phase 2 trials, which involve a larger group of patients with the target disease. Here the focus is on evaluating the drug's efficacy in treating the disease and identifying the optimal dosage regimen. Finally, phase 3 trials involve significantly larger patient populations, often numbering in the hundreds or even thousands. These larger scale trials serve to confirm the drug's efficacy, compare it to existing treatment options, and meticulously monitor for long-term safety. That's all from me. Thank you, Doc.
So now I'm going to be explaining about the stage 4, which is the regulation approval. So once a potential drug successfully navigates the heartless of clinical trials, it must then conquer the final frontier, which is the regulatory approval. This critical stage involves compiling all the data amassed throughout the preclinical research and clinical trials into a comprehensive investigational new drug or IND application. These documents serve as roadmap for the regulatory agency outlining the drug's properties, safety profile, and clinical efficiency. The IND application is then submitted to regulatory agencies such as the FDA in the United States for a meticulous review process. During this process, the regulatory agency may raise questions or identify concerns regarding to the drug safety or efficiency. The drug developer must then meticulously address this concern by providing additional data or explanation. Finally, after a rigorous and often lengthy review, the regulatory agency will render its verdict. It can be approval for the marketing the drugs, a request for further studies, or also a rejection of the application. This decision significantly impact the future of the drugs and its potential to reach patient in needs. That is all from us. Thank you.